Today I'm going to talk about, well, I'm going to give you sort of a little bit of a background to why the traits courses are done the way they're done and how they happened and how they came about, etc., etc. So we're starting from Western Norway, but we're going to end up in Peru eventually. So when we are studying climate change impacts on plants or on vegetation, there's a lot of different approaches that we can use. And a little bit sort of worryingly, when we use these different approaches, we tend to get different answers. So one of the things we can do is, of course, climate change experiments, here represented by an open-top chamber. Those of you who work in Arctic systems will recognize those. We can also do resampling studies. We can travel around and sample studies that have been done earlier and look at the changes over time. That's also something that's a lot done, and that is just showing some very clear changes. And then, of course, we can do observations along gradients in time or in space. And finally, we can use models like species distribution models, etc., to model changes in floras, etc., to changes in climate. If you take all those studies and if you analyze them together, so here's a study from Elmendorf et al. In 2015, what you find is that there is a lot of variation out there. So depending on which region you work in, you get different answers, which is perhaps understandable. But you also tend to get different kinds of answers depending on what kind of studies you're doing. You have more change. The y-axis here is change in number of degrees per change in degrees in the community per degree climate change. So you get more changes in some regions than others, but you also get stronger changes if you do experiments than if you do observational studies, and also if you continue your study for a longer time. So this is sort of worrying. And one of the conclusions that we could draw from this, of course, is that some of these methods are wrong and some are right, and we need to find the best method. But I think it's more useful to think about how models and observations and experiments ask different questions. So if you think about it, for example, when you're modeling, like doing a species distribution model, you have quite long time scales. And what you focus on there is the long-term outcome or the equilibrium state of your community or flora or ecosystem. And the responses that you focus on tend to be ecosystem or biodiversity states and trends. And the questions that you're asking are things like, what are the long-term outcome? What is the state? What is the flux of different ecosystem processes? When you're doing observational studies, then you're working either on instant time scales, or you're working on sort of tens, hundreds, thousands of years, so quite long time scales. Again, you are focusing on equilibrium, but also on changes in the system, and your typical response variables are things like species range shifts, or increasing or decreasing species pools, or changes in the ecosystem state. And the questions that you're asking tend to be things like evolutionary outcomes, local traits, adaptations, species losses or gains, and so on and so forth. When we are experimenting with ecosystems, then our timescales are often very short, or much shorter, and we tend to focus on the transient dynamics, so the ecological processes underlying the changes that we're observing on longer timescales, if you want. And we are manipulating the extant communities, so we're looking at what is happening to the community that we have here now. And so it's the ecological processes and, for example, direct or indirect effects of climate and things like that we are focusing on. I want now to take you, so there is a change in, or a difference in the focus of these different methods. And my argument would be that it's better to try and combine them and think about how we can borrow strengths from one system to another, rather than to try and just focus on one set of methods and one set of studies. So I'll take you on a little tour, first to Western Norway, and then via some of the other traits courses before we end up in Peru. So this is one of the fjords of Western Norway, Sognefjorden. It rains here, it tends to do that. But what is interesting is that we have a lot of climate available in this system. So here, for example, is a mountainside of 1,000 or 1,100 meters. And along that mountainside, we have a temperature gradient of 6 degrees. So here we could do nice gradient studies. And in this fjord system, we have a wide range of summer temperatures and also a wide range of annual precipitation regimes available. So we have a lot of rain out on the coast, and then it's much drier in the inlands. So we use this system to set up a grid of sites, going from the relatively wet areas on the coast to the much drier areas in the inland. And then we also sample different altitudes. So we sampled lowlands where we had relatively warm summer temperatures, and then alpine systems where we had relatively cold summers. And across this entire grid of sites that we selected, we made a lot of climate change experiments. And so we transplanted, for example, vegetation from relatively cold places, like up here, down to some warmer places, and also from relatively dry places to some wetter places, because that's what we expect in terms of climate change in this region. It should get warmer and it should get wetter. And then we did that across all the sites. And the study system that we selected for these experiments were grasslands. They are relatively widely distributed. They are small, so we can do things with them, like we can lift them up and carry them around if we want. They are also relatively fine grained, so you have a lot of species, a lot of diversity on a very small scale. And then they are they respond on relatively reasonable timescales, so we can actually work with them. And then they also have high diversity. And there are climate responses and processes happening in there that we are interested in. And they also have ecological functions and deliver ecosystem services that people are interested in. So we have done a lot of studies and experiments in these systems, and I'm not going to talk about that now, but I'm going to just show you a couple of sort of trait-related things that we have been exploring. So one of the things that we've been finding in this system, for example, and these are two people who have been or are PhD students here and who have also been on the traits courses before, Francesca Jarotzinska on the top and Ragnhild Gya at the bottom. Some of you know her very well already. The rest of you will get to know her on the course. And one of the things that they have been finding is that there are, as you would expect, there are trait trends along these climate gradients. So these are, for example, from the cold blue and to the red warm sites, we see that there is a shift in specific leaf area from relatively low specific leaf area in the alpine system to relatively high in the lowlands, as you would expect. We also see that there are some differences between the graminoids and the forbs. So in the graminoids, you have more of a turnover of traits, and then in the forbs, there is more a change in the variance. So far, so good. But because we do experiments in these systems, we can actually dig into the underlying ecological mechanisms, and we can ask why and how is this system changing in terms of these traits. So one of the things we did, or Jon Gitar, who was also working here, he was also a PhD student here, and one of the things that he did was that he made a simulation model in this experimental setup. So he has some control plots, the ones down here that are not changing, and then we observe how these transplanted plots are moving towards the controls, towards their target sites in terms of species composition. And then we use the simulation model to ask, are these the species composition and are these traits changing as rapidly as you would expect, or are they changing faster or slower? And so what we found was that SLA, specific leaf area, was changing in response to these experimental treatments, or in response to this warming, but it didn't happen any faster than what you would expect by chance. What did change a lot faster was things like bud number and persistence of clonal integrations. So what we found was that clonal traits seem to be driving, or seem to be deciding which species are succeeding or failing in this system, and so the SLA is more, the leaf traits are more trailing these clonal traits. And so in this system, we have done a lot of these different types of experiments. So I was talking about these transplant experiments now. That was the first set of experiments. Then we also did some more experiments to look into the interactions between species. So we did removal experiments. And now Ragnhild is doing transplants and OTCs to look at how lowland species are affecting the alpine community as they move up. And Aud, who you will also, some of you know and the rest of you will meet, is looking into how climate change is interacting with grazing and nitrogen deposition in impacting these grasslands. So one of the things that we have been finding in some of these experiments, and so this is from this FunCamp experiment, this removal experiment, that where we're removing the graminoids to see what's happening to the rest of the community. And this is Siri Olsen who was working there. And one of the things that we're finding is that it matters where you are. So we have this grid of sites from warm to cold and wet to dry, and we're doing these experiments, and it really matters where you are in terms of what kind of answer you're getting. So this is, for example, the difference in population growth rate of these Veronicas. And we're seeing that when we're in a cold place and we remove the neighbors, then they actually do worse than if the neighbors are present. But when you're in a cold place, or in a warm place, sorry, and you remove these neighbors, then they're doing better. So there is this change in the response of this particular species and also of the community to the different experimental treatments depending where we are. And in this particular case, we could link this to survival. So there is a change in survival. They're surviving much better if you're removing the neighbors in the warm places, but not in the cold. And context dependencies like this is something that you're finding quite a lot. Basically, if you search for context dependencies and climate change, you're getting like hundreds of thousands of hits in the databases. So there's a lot of these patterns around. And they're of course a bit frustrating, because what it means is that you can't really generalize. It's difficult to generalize, because what effect you have of climate change and also of the changes in interactions, it really sort of depends where you are and what kind of system you are in. So of course, we want to understand these context dependencies. If we could understand them, then we again could be able to predict even if there is some variation in these responses. And so, yeah, and this is just an example of one of these context dependencies. So this is from Europe. Species are generally moving up the mountains towards the alpine as it's getting warmer. This is an observational study. But the depressing thing is that they're not moving equally fast everywhere. Sometimes some places all the species are moving, some places it's just 20% of them or something. So it's super variable, and it seemed to relate to something like, for example, possibly precipitation or nitrogen deposition or something, but it's not clear. After having done these transplant experiments in Western Norway, we decided that, okay, since we have all these context dependencies, let's scale up. Let's try and replicate this around the world. And so we contacted a lot of people who had transplant experiments from other places, and we also set up some. And so we now have this network of sites where we do transplant experiments. And of course, we're not alone doing this. So I know that some of you are in DroughtNet, and some of you are in all these other different networks of experiments and networks of studies. And that's something that's really sort of becoming very, very important and very popular to do these replicated experiments or macroecological experiments, if you want. Now, one of these sites is in China. And that site happened to be a site where near Mount Gonga in China. So it's one of the easternmost above 7,000 mountains in the Himalayan range. And there, I just have to show you this beautiful sign. So there we have a sister experiment to the transplant experiments that we have in Western Norway. So we have this gradient of sites going up the mountain from 3,000 meters to up to 4,100 meters. And there we do these transplants, as you can very clearly see on this figure. And then we also have some open top chambers and so on and so forth. And so we had this experiment. It's also a relatively wet place, so it's very familiar to people from Western Norway. So Kari Klanderud and Yan Yang and I, we had this experiment going in China. Here you can see our open top chambers. They were quite dirty when I took this picture. And then Brian and I started discussing sort of maybe we should do something here. Maybe we should add something. Maybe we should add some trait measurements, because that was not part of this study. And then we somehow came up with this idea that we could do it as a course. So we brought a lot of students, including some familiar faces, to China, and then we did a lot of different things. We measured carbon fluxes, and we did all kinds of measurements inside these open top chambers. We had our Hello Kitty sitting pad that was very popular. And then we did a lot of different things. And so we did this for a couple of years, these courses in China, and then all of a sudden we had this very nice traits data set. And so one of the students, John, who has been a very repeat fellow, he's been on several of the traits courses, he wrote up some of these results as a paper. And one of the things that we did find, which is of course something that resonates with things that we find in a lot of places, is that so these are the different traits that we measured on our leaves, and this is how much variance in those traits we found, or the percentage of variance that we found on different levels of organization. And so there was some trait variation across the sites, so the black patterns here, but quite a lot of this quite a lot of the variation was intraspecific. So we had within species within sites is the orange one, and then the black one is within species within sites, and then we have some higher order variation. So part of what we found was that there is a lot of intraspecific variation, and also part of what we found is that this varies a lot across traits. And then a lot of other things. When in doing this course, or these two courses in China, we not only found things, but we also learned quite a lot. So one of the things that we learned was that we can collect quite a lot of data in a relatively short amount of time if we are a lot of people. So from those 2 years, we have collected traits on almost 200 taxa, and 100 of them are not in the databases before. So that's 100 new species for trait measurements. So that's kind of cool. We measured almost 7,000 leaves over 2 years, which is more than anybody would want to measure. And then we have a lot, because we have measured many traits from those leaves, we have a lot of data points. And the 600% here is how much new knowledge, or how much of the new, how many more measurements we added compared to what existed from those 193 taxa from before. So we're adding quite a lot of new data. And then we can match all that up with carbon fluxes and photosynthesis measurements and so on and so forth. So all that's kind of cool. We also found out that just managing the field work and the data and the people involved is much harder and much more important than we had first realized. And so we also realized that sort of doing this properly and doing this sort of systematically, and also documenting the data and the data collection and the field work and everything is really important. To the extent that we're now writing up these courses as data papers. We're publishing the data and the entire protocols and all these things, and you will all be part of doing that for Peru. So we ran the traits course in the Ganga Mountains in China twice, in 2015 and 2016. And then we got very inspired, and then we ran it in Peru in 2018, and also on Svalbard up in the Arctic. And then we're going back to Peru now. We have similar kinds of data from Colorado and also from Western Norway. And then we sort of have traits courses lined up in South Africa for 2021 and in Norway for 2022. So of course, as you can see, we're not only collecting one data set, but we're also sort of planning for a comparative data set here. So what happened now? Okay, so we are collecting these comparative data along gradients, along these altitudinal gradients. We always try to sample altitudinal gradients. We tend to be in sort of grassland type vegetation. And then we collect data on vegetation and leaf traits and ecosystem carbon fluxes and photosynthesis and the imagery and so on. We also tag on to existing studies and experiments in these various systems. So as we did in China for these transplant experiments, we try to augment whatever is there with detailed trait-related data. So in Svalbard, we were working in open-top chambers. In Peru, we're working on fire, as you know, responses to fire. And so in Peru, in Wayquecha, and those of you who have looked at the data documentation that is now available to you, know that there is a lot of data and a lot of information out there already. And so this is the basic setup that we have for our plots. We are staying in Wayquecha, and then we have various sites up the mountainsides at Aconaco, Pica Grande, Tres Cruces, Quella Casa, and then also some over sort of on the different mountain further to get some further up sites. And in these different areas, we have some control plots, the gray ones, and then we have some that have been burnt around 10 years ago, and we have some that have been burnt in the last 10 years. So we also have natural experiments in terms of fire. And the questions we're interested in exploring along these gradients are, and in those different groups, are what the trait responses, what kind of trait responses we're finding to altitude and fire. So here we're talking in sort of response traits terms. So how are they responding to these different drivers? We also want to understand changes in the carbon fluxes and photosynthesis and how the traits are affecting or mediating those responses. We are interested in the role of intraspecific trait variability, and if you remember back to China, we saw quite a lot of that there. We're interested in knowing, is it the same level of variability, and what traits are varying and why, etc., and what kind of species. And then we are also interested in attaching this to the drones and the imagery to see if we can scale up and try to understand what's happening on a larger scale than in these plots. And in all that, we're of course exploring this natural fire experiment. So in terms of this, in terms of the comparative aspect, one thing we're interested in when we're comparing all these altitudinal gradients, and we don't have the data from all the trait courses perfectly lined up yet, but one of the questions some of the questions we want to be asking are things like, and this is a paper from Forrester et al. Who were comparing from 2017, and they were looking at grasslands in North America and in Africa along an annual precipitation gradient, and they found this very nice matching pattern that, you know, the gradient was exactly the same in terms of how mean productivity was responding to annual precipitation. But what was really interesting in that paper was that when they started digging into the traits and into the patterns in the vegetation, they found that there were actually quite different trait responses or trait patterns underlying this ecosystem response. And that was very much related to different groups of species or different groups of taxa being dominant along the gradient on two continents. So you can have the same functional response being driven by very different underlying trait patterns. And we're interested in exploring and comparing our altitudinal gradients in these terms. When it comes to fire, and this is again back to Western Norway. So we're interested in understanding how fire drives and time since fire drives both the function and also the trait variation in our communities. So this is Siri. She has also been on one of the traits courses. She will not be coming this year, but she has a drought net site on the coast of Western Norway. And the background for this is that although it rains much more in Western Norway now than it used to, we also have a very much more variable climate, and that means more droughts. So we get a lot of these events where we have really high drought, and of course our flora is not adapted to this, and so we get these quite extreme drought effects. And so what Siri's been doing in her studies, she's been setting up this drought net site with different sort of levels of drought, and then she looks at how the plants and how the vegetation in Western Norway is responding to these droughts. And what she finds, what she finds is that these pioneer things, it's supposed to say recent fire down here, but it disappeared. So when we look in terms of leaf dry matter content, so another trait, we see that these pioneers, so these newly burned sites, they can tolerate any kind of drought. There's no problem. There doesn't seem to be any response in terms of the traits, and this is a relative plasticity index, which is one way we can use to explore the trait responses within and between experiments. But when you go towards these older these sites that have a that are sort of have longer since or later in the succession, so there's longer time since fire, then all of a sudden we're starting to see this stress response in sort of affected by the drought. So the late successional stages seem to be less resistant to drought. So things like this we can explore along our fire gradient in Wayquecha, but then or in Peru. But then of course we need good data both on the intra and the interspecific trait variability in response to the fire gradient. And so those of you who have been looking into the folder that was made available to you yesterday on the data documentation will see that we are quite focused on data documentation in these traits courses. And part of the reason that we are is simply because if you want to share your data, if you want your data to be used, if you want to combine your data with other people's data, then of course it needs to be well documented. Those of you who have tried to combine data from many studies into meta-analyses or other kinds of synthesis will know that a lot of studies just aren't well enough documented that you can use them. They don't give the necessary information. And over the past few weeks, it's also been very clear that I don't know how many of you have heard about this Spidergate, but if you haven't, you can check it out. But at least there's a lot of good reasons for sort of pre-documenting your data and keeping track of your data so that you will be able to prove that your data is valid and good and there is nothing wrong with it, and also so that your collaborators can trust your data. It's also not enough for the data to be open and available and documented. They also need to be you need to be able to interact with the data, to find them, to access them, to operate with them, and to use them. And of course, as we all know, collecting data is hard work. And so we want our data to be as useful and as much used as possible. Then thinking very carefully about how we can make that happen is something that we are quite keen on. So we put the British Ecological Society's Guide to Better Science out on your reading lists, and they are very, very useful indeed. We also, and Aud was leading up this work, we also wrote this paper that is also in your reading list, which is a paper that nobody's supposed to read, or you can read the paper, but don't read the appendix, at least not from the beginning to the end. That will drive you crazy, as we confess. Yeah. But what we did in this paper and what we wanted to do in this paper, what we wanted to think about, how can we, so we have all these different experiments and observational studies all around the world, and of course they're all getting published, and that's all fine. But but if you want to reuse the data, so if you want to combine them with other studies, and that is very important, because if you want to understand context dependencies, for example, we need to understand how and why responses vary across different sites and different regions and whatnot. Then you need those data to be able to be used for these second order outputs, or for this synthesis work. And then of course, if the data aren't available, then that makes that difficult. If the data aren't comparable or incompatible, that is another filter. And so you have very few studies. You end up with very few studies when you're trying to synthesize. So we were thinking that, okay, one solution to this is, of course, that you join these coordinated networks like DroughtNet and all these other different networks, and that of course makes things easier. But with this paper, we wanted to help people set up and document and do their studies right, so that more of these single site and standalone studies can also make it down into syntheses. Yeah, and so here's the paper. And so just wrapping up, yeah, those of you who have kids know that kids start out sort of playing beside each other, and then as they get older and get more mature, they, in the best of cases, start to play with each other. We want to think about how we can make gradient studies and experiments and also trait-based studies work better together. And it's all about sort of making sure that we measure the same kind of variables, and that we measure them in ways, and we record our data in ways so that they can be combined. And sort of having some of the different same knobs that we turn in terms of gradients and extent and predictors and response variables is very important. And so sort of a little list at the end of sort of the various lessons that we have learned. First of them is to be both that you should and how to be tidy and document your data well and be robust in your experimental designs and have open, fair data. And we're going to be talking a lot about this in the course, and we're going to be doing a lot of these things in the course because we have learned that it's a really good idea. We also combine gradients and experiments because they ask partly different questions, and also because it makes us able to explore these context dependencies. For example, by combining these different gradients, also combining gradients and experiments in these different studies. We're going to be talking about study design and how and why we're doing the different designs that we're doing in terms of replicating and assessing both within and among sites, the kind of power that we have in our studies, and so on and so forth. Yeah, I've been talking a bit about sort of combining experiments and adding more experiments and more types of studies into your experiments. And then, of course, joining networks and using common protocols and sharing data is extremely important. Oh, and there's another 5, whatever. Yeah. And when it comes to this this first point, actually you will not have we're hoping that after this course you won't need to just believe us anymore, because we will have explained to you how and why all of these things that we are doing in terms of data documentation is a good idea. Yeah, and that was it. And then I have to say that I do like experiments, in case you hadn't noticed. Okay, then I'll stop this sharing. Okay, so is anybody here? It's very weird to be